This video is about the lactose operon. It's an example of how bacteria regulate genes. So we have looked at this many times where DNA is the informational source that's used within all organisms to make RNA copies. And that's known as transcription, the process of taking a DNA molecule and making an RNA copy. And then that RNA is used to make proteins in the process of translation. So there are many proteins that are needed all the time in certain cells, and so those proteins would always be made. But there is also a lot of proteins that aren't needed all the time and instead are only made when they're needed. And so this process is called gene regulation. So it's the process of turning off and on genes when they are needed, you turn them on, and when they're not needed, they'd be turned off. And so what happens is, when we say a gene is off, that means that the DNA is not being used to transcribe, so transcription doesn't happen. So that means RNA is no longer being made, so that will break down over time and will decrease in its amount within the cell. And then over time, if there's no RNA, then that cannot be translated, and so the protein will diminish as well. And so this is a process that we call gene regulation. And in bacteria, they use a series of operons to achieve gene regulation. And so one classic example is known as the lactose operon. And lactose is a sugar. It's a disaccharide, so it's two sugars linked together, two monosaccharides linked together. And sugars can be used, like lactose, to be an energy source in some bacteria. And so within the Lac Operon, there are three structural genes that encode three proteins that are used to metabolize lactose in bacteria. Since they all are needed kind of collectively to break down or metabolize lactose, they are regulated as a single operon. So what I mean by that is, if a bacteria wants to break down or metabolize lactose, all three of these proteins need to be present. So rather than turning them on individually, they're all turned on collectively within one operon that's called the lactose, or Lac Operon for short. Now, operons are used within prokaryotes, and they can be categorized in one of two ways. So inducible operons, which the lactose operon is an example of. Inducible operons, their default state is off. So that means the operon's off, and something needs to happen for it to be turned on, or the word we use is to be induced, so to turn on or to be induced. There's another category of operons that are said to be repressible, and so their default state is on, so they're always on, and something has to happen in order for them to be turned off, so they need to be repressed. So again, we're going to look at the lactose operon. So what we have is a diagram here of a piece of DNA, specifically the lactose or Lac Operon. And there's several pieces within this DNA that we want to draw our attention to. So the first is known as the regulator, and this is far away. So this dotted line represents it's not necessarily very close to this region. But what the regulator does, it's a region of DNA that is constantly making a protein that's called the repressor protein. So this is going to be an important player that we'll look at in just a little bit. Then there's a region called the promoter. We've talked about this region before. It's important in the transcription of all genes because it's the region where the enzyme RNA polymerase binds. And when RNA polymerase binds, it's able to take a piece of DNA and transcribe it into RNA. So that's called transcription. Now, this is a new piece of DNA that we haven't talked about before. It's called the operator. So the operator is a region of DNA where the repressor protein, so again, flashing back, the repressor protein is always made by this regulator sequence, so it's always present. And where it binds is to this operator region. And then within an operon, we talk about structural genes. So these are the ones that are actually doing the job. So when these proteins are made in the lactose operon, there are three genes here that are used to metabolize or break down lactose. So in the lactose operon, there are these three genes, and they are used to break down the sugar lactose. So here's a question. When would it make sense to make these proteins to metabolize lactose? When lactose concentrations are high or when lactose concentrations are low? So if you want to break down lactose, it needs to be present. If it's not present, it doesn't make sense to have proteins to break lactose down. So the lactose operon will be off when lactose concentrations are low, and then the operon will get turned on or induced when lactose levels get higher or increase. So as we said, the regulator is constantly making this repressor protein. And so this protein is always present. Another protein that's always being made by another gene that is not shown here is RNA polymerase. So these two are always present. And I've color-coded these. So RNA polymerase binds to the promoter, and the repressor binds to the operator. Now, what you can hopefully see is this RNA polymerase, although it's bound to the promoter, is not able to get access to these structural genes. And so currently, these structural genes are not being transcribed. And so we'd say this operon is off. So the repressor is bound to the operator. It's blocking RNA polymerase from transcribing the structural genes. And so if there's no transcription of these structural genes, then there's no RNA. So if there's no RNA, then there can be no protein. Okay, so that's what's going on as the default state. But what happens if lactose becomes available in the environment? So this can happen from a variety of ways. Maybe you put bacteria onto a new media that contains lactose, or whatever the reason may be. And so these blue circles represent now the presence of lactose. Now lactose has a role. We call it the inducer. And so it's going to be the trigger that turns on this lactose operon. And the way it does so is, hopefully you can see there's a little space that's currently empty in the repressor. It was empty in the previous slide because lactose was not present. But now, because lactose is present, it can bind to the site. So lactose binds to the repressor. So again, lactose is known as the inducer because what's going to happen is when lactose binds to the repressor, it causes a change to the repressor, change in shape to the repressor, so it can no longer interact with that operator sequence. And once this space becomes available, you can probably guess what happens. RNA polymerase is now available and now ready to interact with the structural gene because it's no longer being blocked. And so it's going to make RNA, as shown here in pink, and then that RNA can be used to make proteins. And so here are the proteins that break down lactose, and that's exactly what they do. They metabolize lactose. And so that's what they're going to start to do until they've broken down the lactose so much that when lactose concentrations are depleted, this repressor is going to have the site empty again because all the lactose has been metabolized or broken down. And so as you might guess, when the repressor returns to this state, it can now bind to the operator again, and that will turn the operon back off because the repressor is now bound to the operator again.